Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes. Welcome to our tutorial on exploring the regulatory pathways of TP53 gene using ConsensusPathDB human. But before starting this tutorial, first we will know what are we working on. Our investigation is on TP53, often referred to as the guardians of genome. It is a genome that codes for a protein called tumor protein P53. TP stands for tumor protein and 53 refers to a molecular weight of the protein, which is approximately 53 kilodaltons. So P53 protein is a critical regulator of cell division and acts as a tumor repressor. Plays a crucial role in monitoring the integrity of the genome by regulating cell cycle progression, DNA repair, apoptosis, and cellular aging. So when cell experience stress or damage, such as DNA damage or oncogene activation, P53 is activated and orchestrate appropriate cellular responses to maintain genomic stability and prevent the development of cancer. About the pathway, now why we use ConsensusPathDB. It is used to integrate and analyze various biological pathways and interactions from multiple sources. It eliminates redundancy in pathway data, facilitates exploration and analysis of biological pathways, assists in making informed decisions for experimental design and target identification, and lastly supports research in bioinformatics, system biology, and drug discovery. So a little bit of knowledge is always preferable for understanding what we are working on. Now let's start with the tutorial. Open an appropriate browser and enter PathDB and click on ConsensusPathDB. Here you can see we have three databases: human, yeast, and mouse. We will work on human gene. Always use a database which is mentioned in a literature paper. So this database is connected with all of these databases. Click on Search in the sidebar and add the interested gene you want to see the path. Here I will type TP53 and then click on Search. Here we have an array of pathway names. We want to look into the regulation and the expression of TP53. So, we will make an interaction between regulation of TP53 activity and expression. Select it and then click Show Interaction. Here we have a lot of pathways listed involved: regulation, expression, acylation, methylation, and degradation. Select any two. We will click the first, which is association with cofactors and regulation of TP53 through methylation. Now click on the button Map and visualize the interaction. Here you can see a bundle of interaction between each other. If you want to have a knowledge of what you are working with, click on Graph Legend and you will know what is the shapes are indicated of. You can see physical entities color in the description below. The graph is movable. You can move all the graphs and create a understandable pathway. And when you point on the cursor, you will also see the information about that particular gene or protein or compound. Here you can see I'm navigating through all the compounds to see which work is being done with each other. You can see when I move across, you can see integrated databases as well. If you click on the first one, you can see the physical entity properties, all the information and accession number related to all the integrated databases you can find. There are certain compounds which show these dialog box as well. You can edit the pathway as per your liking. Here I clicked on the interaction information and you can see all of its information, where it is cited as well. And you can even see the backlink of interactions as well, what processes are being taken. If you're interested in targeting a particular interaction, you can make connection between them by selecting it, and then it will make a graph upon that site in which you are interested in. Here you can see your interested interaction as well. Anyhow, backing it up, you can see the whole array of interaction which is being taken on behind the TP53 gene. When you click on miscellaneous function, you can even see what type of physical entities and how many of them are in this pathway. If it is very confusing, you can even search your particular physical entity. Unfortunately, there is no availability for exporting it in image file. Anyhow, you can still take screenshot and save it for your research paper or your interest.